Here's the main point of the talk. When a trait depends on strongly opposing forces, then imbalance and disease arise quite easily. Let's start with an example from David Haig's work. Mammalian genomes contain very strong growth promoters, and those strong growth promoters are opposed by very strong growth suppressors. For example, the mammalian gene IGF-2 is a very strong growth promoter. And interestingly, IGF-2 is paternally expressed. What that means is that at a diploid locus, you inherit one allele from your mother and one allele from your father. The maternal allele is turned off and not expressed, and the father's allele is turned on and is expressed. So IGF-2 is a paternally expressed strong growth promoter. And in mice, there are about 6 or so paternally expressed very strong growth promoters. On the other side is a gene such as H19, which in mice is a maternally expressed growth suppressor. And mice have about 5 or so maternally expressed growth suppressors that are very strong. And so overall, mammalian growth then is the opposition of these strongly expressed, very strong paternally expressed growth promoters and strong growth suppressors that are maternally expressed. And humans have many of these same genes. So we have this strong opposition of forces. And in humans, for example, when one of the maternally expressed growth suppressors is knocked out or is not fully expressed, then we have an imbalance because we have an excess of growth promoters that are no longer balanced by the suppressors. And that leads to babies that grow very fast, that have large tongues, they're large babies, they feed excessively, and their tissues overgrow and they're susceptible to many different kinds of cancers and different kinds of tissues. And this is a syndrome known as Beckwith-Wiedemann syndrome. In mice, experimentally, if we knock out one of the maternal growth suppressors, one at a time knocking out one of those suppressors, then we have an imbalance with too much growth promoter, and the babies in the fetus and the placenta are about 30% larger than normal, which is an extreme overgrowth. On the other side, if we knock out in mice one of the paternal growth promoters, then we have too much growth suppressor, and the babies are about 30% too small. So that's a huge undergrowth. So we see that an imbalance arises very easily between these growth promoters and growth suppressors. And when the imbalance arises, we get extreme abnormalities or disease. And so that's the general idea, is this strongly opposing forces are easily perturbed, leading to imbalance and disease. And so the question for the remainder of the talk is this a general principle that might help us to understand other aspects of disease for other traits? And there's no direct evidence for this, but I want to speculate a bit about 2 different kinds of traits that have strongly opposing forces and where imbalance may lead to disease, and ask whether this notion of opposing forces and imbalance then is a general principle that can help us to understand disease more broadly. And the first example comes from immunity. And here's the idea. When a host gets infected, it's very important for the host to trigger its immune response very quickly. There's quite a bit of evidence that when hosts trigger their immune response relatively slowly, then the pathogen and the infection build up and the severity of disease is much greater. So there's a need for speed in triggering the immune response. You need quick triggers. And quick triggers of immunity are prone to errors and making mistakes. Because the trigger is so quick, occasionally there'll be false alarms, and that immune response will be triggered even though there actually is not an actual infection. And so the immune system needs strong negative regulators of immunity that can turn off immune responses when there are false alarms. This is important because an immune response is very costly, and turning on an immune response when there is in fact no infection is a very costly thing. So there's a need for these very strong negative regulators of immunity. So the idea is that strong triggers of immunity are opposed by strong negative regulators. And in fact, the immune response of many organisms exactly has this kind of balance between quick, strong triggers and negative regulators. One line of circumstantial evidence is that there are many gene families of triggers of immunity that are conserved throughout multicellular life. They go back to the origin of multicellular organisms and are conserved, and these are big, diverse gene families. And many multicellular organisms have hundreds of these genes in their genome, and sometimes even thousands of these genes, suggesting that there's very strong selective pressure for these strong, quick triggers. On the other side, there are some conserved gene families that are very strong negative regulators of immunity. For example, in plants, there's an ancient group family of microRNAs that act as negative regulators of immunity, and in particular, they tend to turn off particular family of triggers of immunity in plants. So we get this strong set of opposing forces. Another line of circumstantial evidence is that in mammals, the triggers of immunity often upregulate interferon, and interferon then becomes a signal to other aspects of the immune system to upregulate and turn on. And interferon is known to have many strong negative regulators. Imbalance between the triggers of interferon response and the negative regulators of interferon response often lead to immune diseases. There's been a couple of review articles recently that have been published talking about imbalances in interferon regulation and how those imbalances lead to disease. So there's a variety of circumstantial evidence suggesting that imbalances in the strong triggers of immunity and negative regulators lead to immune diseases. The second and final example that I want to talk about is similar in structure. And the idea is that danger needs to have a quick trigger of fear, a fear response. It's very important to respond very quickly and strongly to signals of danger behaviorally. And that will lead to a variety of false alarms because the trigger is very rapid for the fear response. There have to be negative regulators of that fear response to turn off the fear response. So once again, we have these strongly opposing forces. And again, there's a variety of types of circumstantial evidence. For example, in mammals, the insular cortex of the brain is a locus of certain kinds of fear response. And there are strong negative regulators of the insular cortex fear response, some of which are bodily. They exist in the body and they send signals back to the brain. For example, decelerating heart rate is a signal to the insular cortex to turn down the fear response. And experimentally in mice, when the connection between the insular cortex and the bodily negative regulators of fear response in the insular cortex is severed, then you get imbalance and you get inappropriate behaviors that would be disease-like states. In humans, imbalances in the insular cortex are known to lead to anxiety, addiction, hyperactivity. So this suggests that insular cortex imbalances are associated with a variety of behavioral and psychiatric diseases. Similarly, in the amygdala, there are strong fear responses and strong negative regulators of fear, and imbalances in the amygdala also lead to psychiatric disorders. On the other side, sort of a reverse situation may happen with dopamine regulation. Dopamine increases in response to positive situations relatively slowly, but there are very strong, rapidly acting negative inhibitors of dopamine that can turn off a dopamine response. For example, in the hypothalamus, there are neuropeptides that act relatively slowly that build up the dopamine response. And there are fast-acting neurotransmitters that turn off the dopamine response and inhibit the dopamine response. So speculating a bit, you can imagine in good situations that require positive rewards, you get a slow buildup of dopamine. But if something goes wrong, then you have a quick negative signal that can turn off the dopamine response. And so overall, then we see behaviorally that there are also strong triggers of behavioral responses that are opposed by strong negative regulators. And then imbalances can lead to diseases and psychiatric disorders. So finally, in the last part of the talk, I want to discuss a way that we might test this idea. And to test the idea, we need to reframe a bit the notion of mechanism here with regard to these triggers and negative repressors. The idea is that the trigger is basically a threshold, and the negative regulators also are threshold responses. And so basically what we have are two different threshold responses, positive triggers and negative regulators. And if those threshold responses get out of balance, then we tend to have a disease state. And so this leads to the following idea. And I should also say that many traits have thresholds, but what's special about these traits is that there's a positive threshold and a negative threshold, and those thresholds need to be in balance. And this is where perturbation and imbalance then relatively easily arises, and why I think these kinds of traits are particularly susceptible to disease. And now, trying to test this idea in relation to these thresholds, here's the idea. The intensity of triggers of immunity for infection is likely to vary over space and time as the intensity of parasite attack varies. Also with danger, as the intensity of danger varies over space and time, then the triggering threshold for a fear response or for an immune response then might vary and be retuned by natural selection. And in the same way, we might imagine retuning of the negative regulators. And so different populations are likely to experience different forces and have their thresholds tuned differently. And so when parents from different populations mate, then the thresholds for positive triggers and negative regulators may be out of balance, and this may lead to disease. And so the quantitative prediction is as follows. The greater the genetic divergence between parents, the greater the expectation that there'll be an imbalance between the positive and negative regulators of traits such as immunity or behavior, and the greater the probability then of disease states. So greater separation of parents leads to greater imbalance and a greater probability of disease. And this is potentially a testable idea because with modern genetic technology, it's relatively easy to measure the degree of genetic divergence between parents, and it's relatively easy to score particular kinds of immune imbalances and behavioral imbalances among the progeny. So this would be a testable idea. So overall, I've argued that strongly opposing forces are easily perturbed and can lead to imbalance and disease. And this may be a general principle that can help us to understand evolutionary aspects of disease much more widely. Thank you.